Biology at Temple University, and his lab studies transcriptional regulation mechanisms that link cardiac stress with altered myocardial fatty acid and glucose metabolism. And today he will talk about navigating cardiac metabolism at KLF5 to treat diabetic cardiomyopathy. Kostas, thanks for giving a seminar today, and it's all yours.
Thank you very much for the invitation first, and I would like to commend your initiative in putting together this session with a lot of trainees, and I'm looking forward to your question and discussion that will follow up after the presentation. So let me share my screen and. All right. So the presentation today will be about our favorite protein, which is KLF5, and I will tell you more about it and the involvement of this protein in the regulation of cardiac metabolism as well as diabetic cardiomyopathy. So how did it all start? So like Lale already mentioned, I was at Columbia University as a postdoc in the lab of Ira Goldberg. And at that time, I was working in a project that had to do with sepsis. And our early findings showed that in sepsis, or actually in endotoxemia, which is the right way to describe LPS-induced inflammation, in endotoxemia, J&K kinase is activated, and this leads to activation of a transcriptional factor called c-Jun that eventually led to the suppression of the expression of a protein called ppara alpha. And we noticed that when we inhibited J&K, which prevents activation of c-Jun, ppara alpha expression was increased. So our first question was about c-Jun and ppara alpha and how this transcriptional factor manages to suppress the expression of a protein which is very important for the regulation of cardiac metabolism. So we started with a conventional promoter analysis of ppara alpha, and we came up, when we compared mouse and human promoter, we came up with a conserved potential binding site of c-Jun, AP1 site. And to our surprise, both sites that we detected in the minus 2 kb region of the ppara alpha promoter were overlapping with potential binding sites for proteins that are called KLFs. So at that time, I had no idea what KLFs are, and it seems like it's a big family of transcriptional factors. There are 17 or maybe 18 different isoforms. They have a similar structure in terms of the DNA binding domain, and depending on whether they interact with transcriptional coactivators or co-repressors, they can eventually activate or repress transcription. So we were very much interested in the regulation of metabolism. So initially we started looking, okay, what metabolic pathways are KLF proteins involved in, and you can see here that for 4 cell types or organs that are important in metabolism, there are several pathways that KLF proteins participate. So the first question was, which is going to be our favorite KLF isoform among the 17 different members of the family? So we did an experiment with endotoxemia. So we injected mice intraperitoneally with LPS. We collected the HATs and we ran a whole genome array analysis. And this analysis, which was followed by real-time PCR validation, drew our attention to 2 isoforms, which were KLF5 and KLF6. So then we treated a mouse cardiomyocyte cell line with LPS, and between KLF5 and KLF6, it seems like LPS induced the expression of KLF5. So we decided to focus on KLF5, and the initial hypothesis was that probably KLF5 and c-Jun somehow interact with each other, either physically or functionally, and they suppress the expression of PPAR alpha promoter. So to confirm this, we generated an adenovirus that expresses a constitutively active form of c-Jun, meaning a mutant c-Jun form, which does not need to be phosphorylated in order to be active. And this, as predicted, the overexpression of this superactive, let's say, c-Jun, led to suppression of PPAR alpha expression. But to our surprise, when we overexpressed KLF5, we saw the opposite result than what we had hypothesized. So KLF5 induced the expression of PPAR alpha. At that time, KLF5 was primarily associated with cardiovascular biology through effects on smooth muscle cells, vascular smooth muscle cells, and endothelial cells that had to do with atherosclerosis and angiogenesis. Also, there were a couple of studies associating KLF5 with macrophages and NF-kappa B signaling, and there was a group in Japan, the group of Ryozo Nagai, who is the guru of KLF5 biology, who demonstrated the importance of KLF5 for fibroblast biology, but there was not much about cardiomyocytes. At the same time, regarding metabolism, KLF5 at that time was associated with regulation of PPAR gamma, and this had been shown to be important for adipocyte differentiation and expansion of adipose tissue, as well as for lipid storage in the liver. And also KLF5 was interacting with PPAR delta, and somehow it was involved in the regulation of skeletal muscle fatty acid oxidation, but not much about heart. And there was a study in JCI in 2008 or 9, if I remember correct, from Ryozo Nagai, showing actually that cardiomyocyte KLF5 most probably is not very important for the regulation of pressure overload driven cardiac hypertrophy. And based on the fact that KLF5 RNA copy number is low in cardiomyocytes, people had made the conclusion that probably KLF5 is not very important for cardiomyocyte biology. Despite these findings, we took the risk to make, when I was in Ira's lab, to make a cardiomyocyte-specific KLF5 knockout mice by crossing the KLF5 floxed mice that we got from the group of Jeff Whitsett at the University of Cincinnati with the alpha-MHC Cre expressing mice, and eventually getting the cardiomyocyte-specific KLF5 knockout mice. So, as we had hypothesized, those mice that had lower KLF5 expression, both male and female mice, also had lower PPAR alpha expression. With chromatin immunoprecipitation assays, we confirmed that KLF5 indeed binds on one of the two predicted regions that c-Jun can also bind. And this led us to our final hypothesis, which has been confirmed and published with a paper we published in 2016, that c-Jun and KLF5 actually compete for binding on the same PPAR alpha promoter region. So I already mentioned that KLF5 is important for PPAR alpha, which is critical for regulation of cardiac fatty acid oxidation and fatty acid metabolism. So we started thinking about other setups, other diseases that KLF5 may be important for. So at that, about 20 years earlier, Dan Kelly had published a paper showing that PPAR alpha seems to be important for diabetic cardiomyopathy in humans and experimental animals. And when they made the cardiomyocyte-specific PPAR alpha overexpressing mice, they noticed that there was accumulation of lipids in cardiomyocytes and also induction of cardiac dysfunction. So based on this finding, we wanted to assess potential involvement of KLF5 in cardiac dysfunction that is associated with diabetes. So this is the model we use for mimicking type 1 diabetes. We inject mice with streptozotocin. We follow the protocol of 5 daily injections instead of a single high-dose injection. And about 1 to 2 weeks later, the mice have high blood glucose. And by 3 to 4 weeks after streptozotocin injection, we observe cardiac dysfunction. So here you can see that the mice, after injection of streptozotocin, they increase plasma glucose and eventually develop cardiac dysfunction. And this is both for systolic and diastolic function. So we checked these mice, and as we expected, we observed that KLF5 expression in the heart was increased, and this was associated with increased expression of ppara alpha. And this was 12 weeks after induction of diabetes. So at that time, we had these 3 main questions. First, whether KLF5 upregulation has some sort of clinical relevance with diabetic cardiomyopathy. What is the mechanism that leads to upregulation of KLF5, and whether any change of KLF5 can really account for diabetic cardiomyopathy. So starting from the clinical relevance question, with the help of George Kararigas, who was at the University at the Charité University in Germany at that time, and now is in Iceland. So they measured for us KLF5 expression in cardiomyocytes of isolated cardiomyocytes from patients with aortic stenosis that either had or did not have diabetes. So in these groups of patients, it seems like KLF5 expression is significantly increased. So this is how we started building up the pathway. So in type 1 diabetes, it seems like in both humans and experimental animals, KLF5 is increased. And of course, this is associated with cardiac dysfunction. And the 2 questions we had was how it is regulated and whether it really accounts for cardiac dysfunction. So we did again a promoter analysis for human and mouse KLF5 gene. And of course, there was a long list of potential binding sites for different transcriptional factors, but our attention was drawn by 4 conserved potential binding sites for FOX proteins. And we were interested in that particular group of proteins because it is known that with insulin signaling, besides regulation of GLUT4 translocation to the membrane, AKT phosphorylation also affects FOXO1 nuclear localization. In other words, when AKT phosphorylates FOXO1, it blocks the localization to the nucleus. But when we have insulin deficiency or insulin resistance, meaning type 1 or type 2 diabetes, the lack of phosphorylation of the inhibition of phosphorylation of AKT results to dephosphorylation of FOXO1, and this leads to translocation of FOXO1 to the nucleus and eventual activation of gene expression that is controlled by FOXO1, depending on another set of post-translational modifications that I will not tell you much about now. So in those diabetic mice, we confirmed that phosphorylation of FOXO1 was suppressed, and this was associated with increased expression of KLF5 and ppara alpha, both at the RNA and protein levels. So we did some in vitro experiments to confirm that FOXO1 does regulate KLF5, and so in the first set of experiments, we infected AC16 cells, which is a human cardiomyocyte cell line, with either wild-type FOXO1 or a constitutively active form of FOXO1. And in both cases, it seems like KLF5 is increased, both at the RNA and protein levels. So then we modified a little bit the treatment, and we added also insulin. And insulin activates phosphorylation of FOXO1, which blocks its nuclear translocation. But insulin does not work on the constitutively active FOXO1 because this one is active independent of phosphorylation. So in this case, we noticed that when we added insulin, and FOXO1 was phosphorylated only in the case of wild-type FOXO1, KLF5 activation expression did not increase, but the constitutively active FOXO1 that was not subjected to phosphorylation by AKT when cells were treated with insulin still gave us a high expression of KLF5. So we did chromatin immunoprecipitation, and we confirmed that FOXO1 can indeed bind on one of the predicted regions in the promoter. So the next question was whether FOXO1 inhibition first would prevent KLF5 upregulation and would alleviate cardiac dysfunction in mice with diabetes. So we we generated the alpha-MHC FOXO1 knockout mice that do not express FOXO1 in cardiomyocytes, and we induced diabetes in these mice. So this is wild-type mice with diabetes. These are FOXO1 knockout mice with diabetes. We can see that both at 4 and 12 weeks, they have high levels of plasma glucose. But the FOXO1 knockout mice have significantly lower KLF5 and ppara alpha expression. We confirmed that this also happens at the protein level, and importantly, when we checked for cardiac function of the FOXO1 knockout mice, we noticed that opposite to the flox mice that develop cardiac dysfunction at both 4 and 12 weeks post induction of diabetes, the FOXO1 knockout mice had pretty normal cardiac function. So the next question was whether the protective effect that we observed in the FOXO1 knockout mice had something to do with lower KLF5 expression. So to test this, we did a number of experiments. So the first approach was to use wild-type mice and induce diabetes with streptozotocin injection, and then treat the diabetic mice with either a vehicle or an KLF5 inhibitor. And 6 weeks later, we assessed cardiac function in these mice. So the experiment was that we induced diabetes. We waited for 6 weeks until the mice have already developed cardiac dysfunction, and then we treated the mice with the KLF5 inhibitor. So the KLF5 inhibition worked. So here you can see we don't really know how this inhibitor works, but we do know that it blocks the expression of KLF5. So in the diabetic mice, KLF5 expression, cardiac KLF5 expression was not increased. This is RNA, this is protein, and the plasma glucose levels were still high in these mice. And this is what happened with cardiac function. So here you can see that 6 weeks post-induction of diabetes, those mice have cardiac dysfunction, the same that happens in the flox mice. And when we start the treatment, the mice gradually improve their cardiac function. Again, this is a drug, so a drug can have multiple effects. To confirm that the cardioprotective effect of the ML264 was KLF5 specific, we induced diabetes in the KLF5, in the cardiomyocyte specific KLF5 knockout mice, the alpha-MHC KLF5 knockout. So these mice, like the flox mice, have high levels of plasma glucose, and in the same way like the alpha-MHC FOXO1 knockout mice, the KLF5 knockout mice do not develop cardiac dysfunction. So the next question was, because we had shown that KLF5 regulates ppara alpha, the Dan Kelly's group had shown that ppara alpha is important for cardiac dysfunction and diabetes. So the question was whether the beneficial effect of KLF5 inhibition is accounted for by suppression of ppara alpha expression. To address this, we got the ppara alpha knockout mice, and we subjected them to diabetes. But those mice were not protected from diabetic cardiomyopathy. So we believe that the effect of KLF5 in causing cardiac dysfunction is separate from the effect that ppara alpha has in doing the same thing in diabetes. And to confirm that KLF5 can induce cardiac dysfunction itself, we treated wild-type mice with an AAV virus that expresses human KLF5 under the control of a cardiomyocyte-specific promoter. So we injected the mice, we waited for 8 weeks, and then we measured cardiac function. And you can see here that in 8 weeks, we have significant increase of KLF5 protein expression in the hearts. And those mice without diabetes develop cardiac dysfunction. The second approach to prove this was that we generated an inducible cardiomyocyte-specific KLF5 constitutive expressing mice. So I will explain how we did that. So, we got mice that have RTTA controlled by a constitutively active promoter that has a stop cassette in between. And this stop cassette can be excised when the mice are crossed with mice that express Cre in certain cell types. So these mice that when this cassette is excised, the mice express RTTA, and those mice have a TRE element, meaning a promoter element that can be activated by RTTA when RTTA is activated by DOX. And when this happens, the mice express constitutively KLF5 in the cell type that expresses Cre. So we created these mice that express KLF5 constitutively in cardiomyocytes when the mice are fed on doxycycline-containing diet. So we confirmed that these mice do have higher levels of expression of KLF5 in cardiomyocytes, and those mice, by second week after expression of after activation of the expression of KLF5, have developed cardiac dysfunction, which progresses further by week 4. So it seems that KLF5 can induce cardiac dysfunction itself, but we didn't have a clue about how this is happening. So it's obvious that this was not through ppara alpha. So we started thinking about what may be happening in diabetes, and KLF5 causes cardiac dysfunction. So more than 30 years ago, people had observed that one of the aggravating consequences of diabetes for cardiac function is oxidative stress. So oxidative stress has to do with the formation of reactive oxygen species. There are different types of reactive oxygen species. Oxidative stress starts with transfer of electrons to oxygen, which leads to the formation of superoxide that can eventually lead to the formation of hydrogen peroxide or hydroxyl anion. So we started from that and said, okay, let's see if KLF5 has something to do with oxidative stress. So we used hearts from mice that were on diabetes either for 4 or 12 weeks. And also we compared the hearts with skeletal muscle. So in diabetes, oxidative stress happened in both the heart and the skeletal muscle. This is DHC staining, and that indicates accumulation of reactive oxygen species. So 12 weeks post-induction of diabetes, we see that the mice have increased ROS in both the hearts and the skeletal muscle. So then we use the transgenic mice that do not have diabetes, and those mice that express KLF5, overexpress KLF5 in cardiomyocytes have increased ROS staining in cardiomyocytes, but not in the skeletal muscle. Then we went to the mouse models of diabetes. This is the knockout, and you can see that opposite to the diabetic flox mice that have increased oxidative stress, the knockout mice do not have increased ROS staining in the heart, but they still have increased ROS staining in the skeletal muscle. Remember, this is a cardiomyocyte-specific knockout. So then we check the mice that we subjected to systemic inhibition of KLF5. And in this case, oxidative stress was blocked in both the heart and the skeletal mass. So this gives us the idea that KLF5 seems to be important also for oxidative stress in skeletal muscle. So then we did an in vitro experiment, and with this experiment, we first overexpressed KLF5 in AC16 cells, and you can see that the increase of ROS staining in those cells. And then we treated those cells with the control cells with palmitate or high levels of glucose that which we know that induce oxidative stress. And in all cases, it seems like KLF5 exacerbates oxidative stress in these cells. One of the consequences of oxidative stress is that the mitochondrial integrity is compromised. So we measured with the mitosox formation of reactive oxygen species mitochondria, and this was increased. And we measured mitochondrial copy number. So it seems like the transgenic mice have about 50% lower mitochondrial copy number, and also mitochondrial protein expression was lower in these mice. And this is how we got into the oxidative stress adventure, because, and I'm saying that adventure because there is so many proteins that are involved in the oxidative stress and the redox balance in cells. So we started with a massive assessment of gene expression for almost 30 different genes that encode for proteins that are associated with oxidative stress. And it seems like the most striking findings were that we saw upregulation of NOx2 and NOx4, NADPH oxidase 2 and NADPH oxidase 4, and suppression of 3 different proteins that are associated, that have an antioxidant effect. So again, we checked for the proteins that we picked up from the first screening in all 3 different animal models that we used. And to make the long story short, it seems like KLF5 was changing reciprocally with NOx4. So whenever KLF5 was up, NOx4 was up, or the opposite. So then we checked NOx4 expression in the transgenic mice, and you can see that there is significant increase of NOx4 expression, and which did not happen in diabetic mice when KLF5 was inhibited either in cardiomyocytes genetically or systemically with the KLF5 inhibitor. So NOx4 is a member of the NADPH oxidases family. It mediates the first step of ROS formation by transferring electrons to oxygen. To confirm that KLF5 is indeed a regulator of NOx4, we overexpressed KLF5 in cells, and we observed increase of NOx4, no change in the expression of NOx2. Then we used an adenovirus that expresses SH for NOx4, and we treated cells with a KLF5 and did DHE staining. You can see here that KLF5 increased, like it happened in the previous experiment I showed, ROS staining, which did not happen when we combined KLF5 with SH NOx4 or with aposine, which is a general NADPH oxidases inhibitor. So it seems like NOx4 mediates the pro-oxidative effect of pro-oxidant effect of KLF5. Again, we did promoter analysis. With luciferase assay, we confirmed that KLF5 stimulates promoter activity of NOx4, and this it also binds on one of the predicted regions in the promoter of NOx4. So it seems like KLF5 stimulates NOx4, which can account for reactive oxygen species. And although not very excited about this possibility, because the antioxidant therapies in diabetic patients did not really improve survival or cardiac events, we wanted to test whether oxidative stress, at least in mice, accounts for a cardiac dysfunction when in cases that KLF5 is upregulated. So when we used a Ross scavenger, a drug that we got from our collaborator at UPenn, Melpo Christofidou, and we treated the mice with diabetic mice with this drug. We confirmed that this suppressed, at least partially, the DHE staining in the hearts, and it completely blocked the oxidative stress in skeletal muscle. But when we checked cardiac function, we only see a partial improvement of cardiac function in diabetic mice that were treated with the ROS scavenger. We also treated our transgenic mice with the ROS scavenger, and in this case, although again we saw complete inhibition of ROS formation, again the cardiac function was only partially improved. So we said probably there is something else that is going on there besides oxidative stress. So we are a lipid metabolism lab, so we did lipidomic analysis in all mouse groups that we used for this project. And when we did hierarchical clustering, we noticed that, interestingly, the transgenic mice grouped together with the diabetic mice, while the mice that we had applied inhibition of KLF5 expression grouped with the healthy mice. So we focused on two toxic lipids, ceramides, diacylglycerols, because these lipids have been associated with complications in diabetes. And it seems like the diabetic mice had the same profile with the transgenic mice only for ceramides. So you see here that ceramides are increased in the hearts of diabetic mice. This is blocked when KLF5 is blocked. And transgenic mice also have increased ceramide, cardiac ceramide content. But for diacylglycerol, although diabetic mice have increased diacylglycerols, as it has been shown in the past as well, the transgenic mice did not have the same effect. So we decided to focus on ceramides, and ceramides are formed through three different pathways. One is the de novo ceramide biosynthesis pathway, which is catalyzed by an enzyme called serine palmitoyl transferase, or SPT, that has two different subunits, SPTLC1 and SPTLC2. The salvage pathway that is controlled by ceramide synthesis is another way to form ceramides, and the sphingomyelin pathway, which is driven by sphingomyelinase, is the third pathway to form ceramides. So the first approach was to see, when we overexpress KLF5, which one of the pathways, which one of these important enzymes may be activated. It seems like ceramide synthase is not activated by KLF5, the opposite I would say, and same for acid sphingomyelinase. But for SPTLC1 and SPTLC2, which are the subunits of the enzyme SPT, of the SPT enzyme, it seems like KLF5 induces the expression. We confirmed that this also happens at the protein level. So here you can see that in cells that overexpress KLF5, SPTLC1 and SPTLC2 are increased. We checked the transgenic mice, and SPTLC1 and SPTLC2 are also increased there. We did a number of experiments. We confirmed that KLF5 stimulates activation of both SPTLC1 and SPTLC2 promoters. The promoters of these genes share about 50% homology, although they are two different genes. And we confirmed again that KLF5 can bind on the promoters of SPTLC1 and SPTLC2. So to assess whether ceramide biosynthesis is important for the cardiac dysfunction we see when KLF5 is activated, we use our transgenic mice, and we treated them with, we fed them on a diet that contains both doxycycline and myriosin, which is an inhibitor of the SPT enzyme. So we confirmed that ceramide content was suppressed when we did the treatment with myriosin. And to our surprise, we saw that there was a complete restoration of cardiac function, complete prevention of cardiac dysfunction in the mice that, in the transgenic mice that were treated with myriosin. We checked 2 and 4 weeks after the beginning of the treatment. So to summarize, type 1 diabetes induces FOXO1. I didn't have much time to show you, but in the paper we just published, we also showed that FOXO1 can itself induce ppara alpha expression. FOXO1 regulates the expression of KLF5. KLF5 stimulates both NOX4 and SPTLC1 and SPTLC2 subunits of the SPT enzyme. And this leads to a combined accumulation of reactive oxygen species and ceramides that lead to cardiac dysfunction. And we showed that by blocking FOXO1 or by blocking KLF5, we can prevent cardiac dysfunction. And by blocking ROS formation, we partially improved cardiac function. And when we treated the transgenic mice with ceramides, with a miriosine that blocks ceramide accumulation, we saw complete restoration of cardiac dysfunction. Interestingly, when we checked for a potential link here, we noticed that when the mice were treated with miriosine, also oxidative stress was alleviated. So there may be some connection here as well. So that's it. I would like to thank all the people who have contributed in this study. The work you showed is primarily the work of Ioannis Kyriazis, who was a postdoc in the lab and is now back to Greece. And the last part for the ceramide pathway is the work of Matthew Hoffman, who's an MD PhD student here at Temple and is now back to the medical year 3. So he is working with real patients now, not mice. And Dimitra Paliura, who's a visiting graduate student, was a visiting graduate student in the lab. She just went back to Greece, and she also helped Matthew with his experiments. So thank you very much.
Wow, Kostas, this was a great story.
Thank you.
Really amazing, both genetic and, inhibitors and really clear pathway, having multiple downstream effectors. Really cool story. And I guess it's, I saw that it's going to be published soon in Circ Research.
Yes,
That's really amazing.
The diabetes story has been accepted. It will be out next month in Circulation Research, and the ceramide story was just accepted by Circulation, and it's actually online already.
Yes, yes. Wow, that's a really cool story. Thank you.
Thanks.
Okay, if you have questions, please use the raise your hand function. And I see that Mabruka already has a question. Mabruka, go for it.
Yes, really very beautiful story. So I know, trainees, we will have plenty of time with you after this seminar, but I cannot wait to ask you the questions. So before anyone will steal my questions. So I know you showed KL5 can go into simulation and post-translational modification, correct?
Yes.
And we know that ROS, and they have playing a role of modifying multiple protein functions. So could be it's the other way around. So in other words, have you tested the post-translational modification effects of ROS on KL5? And this could be perhaps an explanation why you also see correlation of loss of expression.
So this is one of the current directions of this project. So right now we are pursuing with a group in California proteomic analysis first to see what regulates the regulator. When we do that for different reasons. One reason is that because we want this eventually to transform to a therapy. So we know that transcriptional factors are not are not druggable targets. So we want to see it's not only potential effect of oxidative stress. So for KLF5, there has been a number of post-translational modifications, and we're talking about phosphorylation, we're talking about nitrosylation by inos, we're talking about sumoylation, we're talking about ubiquitination. So we want to find what are the PTMs that KLF5 is subjected to in diabetes, and see if we can drug the proteins that regulate that. So I don't have an answer yet to your question, but this is the direction we're now pursuing.
Thank you very much. I'll follow up with that.
Okay, I'll ask the next question. Costa, so you showed really beautiful data with type 1 diabetes STZ model, right? And it works through FOXO1. So in theory, in type 2 diabetes, right, where we have a bigger population, FOXO1 should also be in the nucleus, right? Have you ever tested whether this same pathway also, at least in insulin resistance, is getting activated. What do you think about? What are your thoughts on type 2 diabetes?
Thank you, reviewer one. So this is the question we got from the reviewer, and we actually did a high fat diet treatment for 40 weeks, and we confirmed that KLF5 is increased in type 2 diabetes as well.
I see. Okay, so it is.
We didn't pursue all the pathways, but this is what we aim to do in the next step of the project in the grant.
That's really cool. And also one more note, then I will let Ada to ask her question. Mimo Achille is the world's expert on FOXO1, and he is developing some new FOXO1 inhibitors for treatment for type 2 diabetes and metabolic diseases. It would be nice to see whether those FOXO inhibitors affect cardiac dysfunction, right? Diabetic cardiac dysfunction in the future. That would be really cool.
I definitely agree. And the virus that we use, we got from Domenico Achille.
Oh, okay.
So Ira had got these viruses when I was at Columbia, and we now use them for this project too. And actually, there is another paper that was published, I think about 10 years ago by Joe Hill's group, showing that the cardiomyocyte-specific knockout FOXO1 mice are also protected from type 2 diabetes as well, in terms of cardiac function.
Cardiac dysfunction, right? Yeah. So I think he has a company that is, making new specific FOXO inhibitors. Okay, Ada, it's your turn.
Thanks. So there is increasing evidence. It was first shown actually in the heart that macrophages help cardiomyocytes clear defective mitochondria, and this way inhibit or prevent cardiac dysfunction. So I wonder if you are going to look at this, whether the crosstalk of cardiomyocytes and maybe heart macrophages are different in the KLF5 knockout conditions, or maybe KLF5 has something to do with that whole process.
So that's a good idea. I mean, in my lab, we don't do much work on macrophage biology, but it's a good idea, and we have actually we're actually aiming to see what the role of KLF5 may be in myeloid-derived cells. And because it has been shown that KLF5 actually stimulates NF-kappa B signaling. And we have generated a myeloid-specific KLF5 transgenic mouse line. And we are now working, but in a different setup. It's ischemia reperfusion in cardiac injury. But it's a good idea. What you're suggesting is a good idea. We haven't pursued that.
I have one more, not a question, but so the link between KLF5 and ceramides is really interesting, right? And I guess this is novel that you just developed. And I know you're really heart-centric, but do you think this could be applicable to, since ceramides are important for, metabolic dysfunction, right? In the development of insulin resistance, do you think this could be used in other tissues like liver, adipose, or a whole body KLF5 and ceramides regulation for metabolic diseases in the future, perhaps?
We have almost got the hepatocyte-specific KLF5 knockouts, and this is one of the things we want to test.
That's great.
Also the skeletal muscle specific, the skeletal muscle specific transgenic. So we want to see, because you notice probably that when we apply the KLF5 inhibitor systemically, we blocked oxidative stress also in skeletal muscle, in diabetes. Right. And given the connection that I briefly mentioned at the end of my presentation between ceramides and oxidative stress, I mean the ROS accumulation, it will be very interesting to see what is happening in the skeletal muscle.
Right. Yeah. The metabolic profile. Okay. I guess Esther has a question. Esther.
Yeah, I cannot raise my hand. It was a beautiful story. I really enjoyed it.
Thank you.
It's a bit outside my field, so I may ask a stupid question, but I'm
I
trying
can, if this makes you feel better, I can tell you that when I was a postdoc in Ira's lab, we were working on filing a patent, and on the project we were working at that time, the best question and the best experiment came from a lawyer. So feel free to give a great idea.
Well, I'm not a lawyer either, so but let's see what the question is. So I'm trained, partly trained as a pathologist, and when patients have like high ceramide levels, or we hear the word ceramide, then we always think of these storage diseases like the Gaucher disease, Fabry disease, and they often also have like abnormalities in the heart. So if you have high ceramide levels or high sphingolipid levels, or you miss glucocerebrase, then you get these really weird cardiomyocytes. Did you see anything of that in your mice? So like they have like swollen lysosomes or like really fatty degenerated cardiomyocytes? So.
We haven't checked. We have not checked.
Okay, yeah.
But the connection between ceramides and cardiac function is very interesting. Many years ago, I was fascinated by something I read on a drug. I think it's doxorubicin, and this drug actually, it causes cardiac dysfunction.
Yes.
And interestingly, the drug, when SPT was inhibited, when the drug was given to the mice that do not express SPT, the drug lost its anti-tumor activity. So definitely the effect of doxorubicin in the heart has to go through ceramides, and ceramides alter the content of the lipid content of the membranes. They have been associated with ER stress, probably because of effects of the ER membrane. So it's a great idea. We just haven't checked it.
Okay, yeah, thanks. Great story.
Thank you.
Okay, I guess there are no more questions, but Kostas, this is a really great story, and congrats on the recent published circulation and the upcoming research paper.
Thank you.
And thank you again for giving this amazing seminar today. And I guess there will be trainees networking session afterward.